So now that we know how DNA is actually coded into RNA and into amino acids and into proteins, we can actually look at the changes that occur in this. When you have a base change and it accidentally creates basically a typo, we are going to call that a mutation. So there's a couple different types of mutations you can have. For example, say you're trying to type the word computer. Say my finger doesn't quite hit the C and I hit a key next to it, let's say V. I type in vumputer. There's the typo. My C got switched out or substituted for a V. Or if I'm going really fast, maybe I add a letter. Maybe I hit the R at the end twice. Now I have computer with two R's. Still a typo. Or maybe I'm going extra fast and I miss the C altogether. And I type in umputer. Another typo. DNA, when it's being replicated and created, can go through similar typos. If we figure this is my original, this is what I'm trying to get, I can actually have 3 different types of mutations or typos in my code, in my DNA's bases. I can accidentally make a substitution where I switch a letter out for another one. I can insert a letter where it wasn't supposed to be, in this case that second R, or I can accidentally delete a letter entirely, just like I deleted the C in that very last one. So these are the 3 mutations that we're going to talk about for DNA. Let me show you an example with actual code. Here are 2 codons, ATC and CGC. If I were to do a substitution, I've switched out the T for a G. So my mutation occurs right here. I've only done 1 base. Notice I still have 6 bases in the first one and 6 in the second. I haven't added or deleted any bases or nucleotides. I still have the same number. So I must have just switched one out if I'm getting something different. Now let's do a insertion. I've inserted a right into this spot in between the T and the C. Because of that, it also bumped my letters down 1. That's why I have this lone C hanging out at the end. This is kind of gibberish at the end. It's not really going to code for anything with our amino acid codon wheel. It's just a random C, a random base. We've also changed pretty much everything after that mutation. We've changed all the letters after, so this can really shove everything down and mess stuff up. So insertion is pretty bad. And then the same thing for deletion. I bumped that A out and I shifted everything this way. So now I start with the T, C, C, G, C, and then I have this weird kind of empty spot here that also messes up that last codon. So deletion will mess it up just as much as insertion. So quick overview of these 3. So here we go. We got substitution on the left. I switched out my T for this G, so I just changed 1 base. Insertion. I took this A and I put it in right between this T and C, so it bumped everything over that way and added this weird C at the end. Deletion, I took out the T and it bumped everything this way, and now I have just 2 letters where I should have had 3. So there are actual names for these 2. You'll notice the substitutions on the left, insertion, deletion are on the right. That's because those are the 2 categories we care about. When I do a single substitution where I change a single base, I'm actually only changing 1 point of the code. So this is called a point mutation. I end up with the same basis. I got 3 here, and I got 3 here. I just changed 1 point. Over here, I have actually shifted the frame. And I'll explain what I mean by frame in a second, but basically, I have shifted the frame of the letters I look at because I'm looking 3 at a time. We know the ribosome reads 3 at a time, which is 1 codon. I've actually shifted where I'm looking to find my codon. This is called a frameshift mutation. And both insertion and deletion are part of this category. Insertion, you'll notice, I'll go from 3 to 4 because I've added and inserted an additional nucleotide. Deletion, I went from 3 to 2 because I've deleted and removed a nucleotide. So here's what I mean by frameshift. Let's say this is my DNA sequence. I'm going to use a word that is easy to recognize so you can actually see how messed up it gets. So we'll use cat. So our sequence is 2 codons, CAT, CAT. The frame right here is what my ribosome reads with. It's going to read 3 at a time. So if I'm going to decode this into RNA, and then my ribosome is going to read it, everything's going to be done in 3s. So I'm going to read CAT cat, CAT cat. However, when I have an insertion or a deletion, it actually moves this purple frame. So let's say this is my mutated G. It accidentally, through the typo, gets added over here at the front. So now we're reading it like a book. We got to read the first letter first. So my DNA decodes to RNA, and then my ribosome reads the first 3. So it's going to actually shift the frame to here. So now it's reading GCA, TCA with this random T at the end that doesn't help us. So now instead of cat cat, which was a good, code, it's gikatikata, which is nonsense. So I've really messed this up by shifting my frame in one direction. That's why it's called a frameshift mutation. Our point mutations are going to mess stuff up, but it's not going to be so bad sometimes. Sometimes a point mutation won't even mess it up if it changes one little base. A point mutation can be workable. It usually won't be a devastating effect. So for example, say I switch this C to a B. I now have bat cat. Alright, well that's not what I initially wanted. I wanted cat cat. But bat cat, I'm like, okay, I guess that's another word and it's another animal, so I can work with bat cat. So sometimes a tiny mutation like that might not make that much of a difference. We know when we add this G, that is going to make a significant difference from reading cat cat to this other garbage. This is why it's called a frame shift, and they are much more devastating. Substitutions can be bad, but they can also be tiny little changes that help diversity. Insertions and deletions are usually going to put a big problem in your code, especially if they're early on. If they're early on and they shift everything to the right or everything to the left, that's going to mess up every single codon from the point of mutation on. That means it's going to mess up every single amino acid it codes for after that weird insertion or weird deletion, which, as we know, will make a completely different protein. So what does this mean for the population? Because now we know about mutations, how does this affect diversity in people? And this is called population genetics. Population genetics is just looking at frequencies of diversity in alleles and mutations within a population. Remember, a population is one species, so that could be like all the humans, all the birds, all the dogs, whatever. There are 5 ways that populations actually evolve. First of all, you can't evolve. As an individual person, the DNA you start with is the DNA you die with. You can be a blonde person and dye your hair bright blue, bright green, whatever. Your kids are not going to get that trait. They're going to get your DNA from the sperm of the egg. So whatever DNA you started with, no matter how you better yourself or make yourself worse throughout the course of your life, the DNA does not change. So you can't evolve. Populations over time will evolve through these 5 methods. And since a lot of these are review, we don't have to go into them a whole bunch, but first one is natural selection. We know what this is. Nature selects the stronger, more fit traits based on their ability to get to reproductive age and reproduce better than the other stuff. If you have a trait that lets you breed 100 times versus 1 or even 0, that trait will probably increase because it's a better trait and you're able to spread those genes throughout the gene pool and the environment. Recombination. We did this with the Punnett squares. When you have multiple alleles and you have recombination and jumbling of genetic info and alleles and traits, sometimes you're going to get differences in frequencies, which is going to cause populations to slowly evolve. They can get more of one allele one generation, then that allele takes over, they can have something go the other direction. But recombination is largely because you can have recessive and dominant traits. Gene flow, also review, we know goes from one place to another. This is black beetles heading over to red beetles to live on their island for whatever reason. This can mix and it can change the frequency of alleles in gene pool, and over time that population will evolve because we've flowed genes to another place. Genetic drift is another one we've learned about, but it's more random. This is the meteorite for the dinosaurs. You're taking a random event, fire, something that wasn't like, okay, you can't survive this. This is just bad luck, poor you. And it's going to kill certain things regardless of their trait. So if you have 6 red beetles and 2 blue beetles, and a fire kills 3 of the reds, we now have a 3-to-2 ratio, which makes it look closer to almost like a 50-50 split. If you saw the population in step 3, you'd be like, oh, red and blue are probably just as fit because they're almost the same amount. But in reality, red was 3 times better than blue. A genetic drift just randomly knocked out 3 of my reds. It's bad luck. But genetic drift is random and can influence population, so it has to be a fairly big occurrence, or it just has to affect a very specific part of the population. So all those are review, and you're probably wondering why we're going over this again, because the 5th one is what we just learned. And it is the final piece to the evolution of populations puzzle. Mutations. Mutations are a natural typo. Remember, typos are just typos. They're gonna happen constantly. Think about how easy it is to make a typo in the word computer, which only has 8 letters. Now imagine DNA with millions and millions and millions and millions of letters and codes, all trying to be in the same order. The human body is very efficient at a lot of things, but it's too much. It's too much to not make a typo. It's ridiculous. So typos are going to happen. If they're bad typos, they don't reach reproductive age, then that typo gets eliminated from the gene pool through natural selection. But they still had that typo. So the diversity and the evolution of population can be influenced by the initial mutations. And all these things work together. All of these will work as kind of one big force. It's not going to necessarily be all natural selection, all drift, all flow, all mutations. These are all going to be constantly happening to shape the populations. And over time, the idea is that the population becomes stronger and evolution does its job, and now that population is more durable and more able to survive, because if a population goes extinct, like our stupid friend the dodo bird, or some other dumb animal that, I mean, look at this stupid thing, like why would this thing survive? Then they did not get enough diversity in time, and the environment eventually got to the better of them, came in, killed them off, and they couldn't withstand it. They didn't have any strong dodo birds that could withstand all of the environmental and external factors, and that was that. And it's really kind of crazy that we even exist at all when you think of all the nonsense that had to fall into place, all the things that had to go exactly right for humans to even become a thing. I mean, even weird stuff like lactose intolerance. Like if you're lactose intolerant and you can't drink milk, you're actually the normal trait. Everybody else who can drink milk is a mutant. We used to have the enzyme that broke down lactose in milk kind of die out as we weaned off of breast milk. But just through the craziness of natural selection and maybe some genetic drift and some random droughts and occurrences, it became such a benefit to be able to drink milk through your lifetime that now became the norm. So everybody thinks that people who are lactose intolerant, oh, they must have something wrong with their digestive system. When in fact, that's the normal digestive system. People who can drink milk are the freaks. They're the ones who can actually break it down for their entire life, even though they're not drinking breast milk anymore. So when you think about it, humans as a whole are actually the luckiest species on this planet.